Notice the stark white line of salt crust baking into this cracked earth. Driven by aggressive irrigation, salinization has degraded 30% of agricultural lands. This rapid phase of salt stress triggers physiological drought, the first strike in a two-front war. In this animated cross-section of a root, we see how an osmotic gradient works. High soil salt concentrations exert a strong pull, actively drawing water molecules out of the root tissue. The plant loses moisture even if the soil is completely saturated. Salt's initial assault is mechanical, subjecting the plant to immediate, invisible dehydration long before any chemical poisoning begins. As internal water pressure, or turgor pressure, suddenly drops, the plant triggers an instant survival response. It halts its vertical growth and allows its cellular structure to shrink slightly to conserve mass. To execute this stunting, the plant downregulates essential cell cycle genes like cyclin. This chemical shift slows both the division and the physical elongation of its cells. Simultaneously, the plant forces its stomata, the microscopic pores on its leaves, to close completely, trapping internal water before it can evaporate. However, closing those doors blocks the plant from taking in carbon dioxide, causing the rate of photosynthesis to drop. The plant survives the initial shock of salt exposure, but this survival requires the complete cessation of growth and energy production. Over the following days, the slow phase of the attack begins. Toxic sodium and chloride ions penetrate the root channels and flood the plant's internal systems. This side-by-side molecular comparison shows a case of mistaken identity. Sodium is chemically similar to potassium, a mineral essential for protein synthesis. Due to this similarity, sodium hijacks the plant's HKT1 transport channels, starving the plant of potassium and creating a toxic internal ratio. The accumulating toxic sodium triggers massive oxidative stress. It shreds the architecture of the chloroplasts and blocks the enzymes required for fixing carbon. The plant's internal systems shift from a defensive pause to a state of active collapse, as toxicity dismantles essential metabolic machinery from the inside out. This schematic maps the plant's last defense: exclusion and compartmentalization. Sensors SOS2 and SOS3 detect sodium, activating the SOS1 pump to force it back into the soil. Remaining sodium is pumped by NHX transporters into the vacuole, safely away from fragile chloroplasts. Survival is now a desperate race. Microscopic pumps must bail out toxic ions faster than the saltwater can flood the system. Our vital agricultural crops are largely glycophytes, or salt-sensitive species. Under prolonged stress, their SOS pumps fail and their vacuoles overflow, leading to plant death. Look at this fleshy Salicornia plant thriving in a highly saline environment. It belongs to a group of naturally salt-adapted plants called halophytes, which survive by supercharging these exact cellular defense mechanisms. Halophytes survive the initial osmotic shock by producing massive amounts of osmoprotectants, like proline and sugars, to perfectly balance their internal water pressure against the salty soil. They survive the toxic phase because their genomes possess extra highly active copies of the SOS1 and NHX genes, allowing them to pump and store sodium effortlessly. Thriving in pure seawater is possible through a stronger, scaled-up version of the same biology our crops already possess. Salt will always attack first by stealing water through osmotic shock, and second by poisoning the mineral pathways. Agricultural biotech is currently mapping the supercharged defense systems of halophytes onto our most vulnerable, high-yield crops. Decoding and transferring these genetic defense mechanisms provides the tools needed to secure the global food supply